I'm Tom Warner. I'm a professor of clinical neuroscience in University College London. I'm a clinical neurologist, but half my time is doing research, a lot of it into dystonia. I've been involved in the field of dystonia for probably about 22 years now. I was first introduced to it as part of my PhD when I was working with Professor David Marsden and Anita Harding at the Institute of Neurology. And my role was to look for genes in dystonia, so I had to travel around the country in the UK, visiting families, examining people, getting to meet them, which was a fantastic experience to see the whole spectrum of dystonia. And then I worked in the lab trying to map and find genes, and that was really my first introduction. And really that's when I got the dystonia bug. At the time I did my PhD, we didn't know any genes, and now there are 22, 23, possibly one more coming out, which affect covering lots of different forms of dystonia. And one of the things I realized as I went through my research is that it's all well and good finding these genes, but the next step is to find out what they do. So in the last 10 years, the work that I've been doing and my group are doing are focusing on actually what happens in nerve cells, in the neurons, what these genes actually do when they're working normally, and what they do when there's a problem in the gene which leads to these genetic forms of dystonia. And the hope from studying these quite rare genetic forms of dystonia is we're going to learn mechanisms which we can then extrapolate to all the other forms of dystonia and hopefully lead us in the right path of finding better treatments. So can we prevent forms of dystonia where there is an abnormal gene? At the moment, no. But in the field of genetics, that's obviously one of the ultimate goals, and there's a huge amount of research at looking at ways of perhaps switching off the bad gene. And we know that even for dystonia, we can do this in cell models, in cells grown in tissues. We know it's possible to keep the good gene working normally and switch off the bad gene. And certainly in other genetic diseases, that's beginning to translate into possible treatments. For dystonia, that's going to be trickier because we are still struggling about exactly which parts of the brain we should be switching genes on and off, and it may be we'll have to deliver a way of switching off the bad gene to a much bigger group of cells in the brain, so that's going to be technically more difficult. But either switching off or modifying the way that bad gene works would be an important way, and certainly is an area of great research at the moment.